Thank you for coming. We are going to talk about immune repertoire profiling. And as an introduction to immune receptors, it's a rather complex genetic system created by nature, as illustrated here. T cell receptors, B cell receptors, and soluble form of B cell receptors are organized very similar way with CDR3 region recognize foreign antigen. And problem to study this system is really enormous complexity of immune receptors in the human body. There are several hundred million of different T cell receptors and B cell receptors constantly present in circulation, for example, and they are responsible to a completely different set of the antigens. And from genetic point of view, it's the most complex genetic system created by nature. And how to really to study all this enormous complexity. And what does it mean? 100 million of different T cell immune receptors. It's at least 10,000-fold more than number of the human genes in the human body. You realize, when we are talking about expression profiling is nothing in comparison with the complexity what we need to study here. So the first question, how to study this enormous complexity. We are going to discuss it briefly. Second problem, we need to understand that this system is universal diagnostic and therapeutic system created by nature. So goal of this system really to discover any foreign antigen or pathological antigens in the human body and try to fix it using completely combination of innate adaptive immunity and some multiple other immunological mechanism. How to do it? So if we think about it, it's really this diagnostic system, it can be a universal diagnostic system for any human disease. And why nobody is using it right now? Because, again, problem in enormous complexity of this system. And second, we don't have really direct link between sequence of immune receptor, which is rather easy to identify, and specific antigen which is recognized. As soon as this problem will be solved, and right now a lot of scientific efforts really targeted to solve this problem. In this case, we all know, okay, here's a sequence of immune receptors, and here's what pathological antigens which is recognized in the human body. So we are going to discuss three issues today, which I already listed right now. And to study this type of the studies, we developed three types of assays shown here on this slide. And you can see here on the left, we developed adaptive immune receptor profiling assay, which allow you to profile all your adaptive immune receptors in the single reaction. We developed genome-wide expression profiling assay, which allow you to profile expression level of all 19,000 protein encoding genes, and some combination of receptor profiling and expression profiling in sorted cells or immune cell fractions at single cell level. And all these technologies based on so-called multiplex PCR, where you design set of the primers which allow you to amplify specific region of the mRNA or DNA, and after that use next generation sequence in order to get quantitative information about expression level or copy number of specific target in the biological sample. And here's the example of AIR assay. And what good in this assay, we design primers which allow to amplify all 7 chain of T cell receptor or B cell receptor in one reaction if you use RNA as a starting material. So technology incorporate, we design primers only against functional isoforms. It also can be run directly in cell lysate. Very often you don't even need to purify RNA or DNA. You can just use cell lysate for this reaction. It's also quantitative, and it's allowed to convert read number to number of the starting template molecules what you use in the assay. It's allow you to quantitate number of, for example, the T cells or B cells if you use DNA, or number of RNA molecule which present in your sample. And it's integrated with Mixer software package, which one of the best software package right now for immune repertoire profiling, data analysis. Let's start from something simple, because I'm going to tell you about technology. And this one is how are this data quantitative, and can we quantitate immune receptors in any biological sample. Here's the example how it look. If you take, for example, 50 nanogram of total RNA from BBMC or whole blood sample and try to run this assay, you can see you can generate rather equal representation of heavy and light chain for both T cell receptor and B cell receptor. You identify approximately 100,000 of different clonotypes with the different abundance level. The number one question, okay, if you repeat this experiment 3 times, what you will see? And I can answer is very simple. You will not get exactly the same result. So you will get something like this one shown on this slide. It's slightly more complicated, but if you can see here on the y-axis, it's starting number of the molecules what present in biological sample, 50 nanogram of RNA, for the each clonotype would identify. And clonotypes here you can see sorted from the most abundant, where you can see started from more than 1,000 mRNA molecules to 1, yeah? And what important to understand, this one experiment in triplicates. You can see here reproducibility in triplicates. You can see at least 95% of the clonotypes amplified from single mRNA molecule. What does it mean? It means that majority of clonotypes which are present in the human blood are present at extremely rare naive cells which are not really proliferating and they just circulating there. So this means majority of clonotypes will be detected. It's rather obvious from this slide. If you try to do the quantitation of any immune receptor profiling data, you need to start with the clonotypes which present at least 10 molecules in the sample. In this case, at least you can get some quantitative data. And it means that number of the clonotypes, what you usually can quantitate, it's around maybe 500 to 1,000, not more, from 100,000. And if you try to focus only on the most abundant clonotypes, like shown here, you get rather reproducible quantitative data, where you can really, similar to expression profiling, you can say, okay, it's twofold enriched, activated, for example, these specific T cells or B cells. And you can generate this data using RNA, as you can see on the left side, or on the using DNA. It's rather obvious that DNA is less sensitive because its copy number in the cell, it's at least approximately 20-fold less than copy number of the RNA molecules. So that's why you need significant amount of the DNA to generate similar sort of that quantitative data. And it's usually in the range 5-10 micrograms, which is really not very often available for, especially if you have small number of the samples with a small amount of the cells. Second question, okay, you identify this type of the do quantitative analysis of different immune receptor clonotypes. The question is, how are you going to identify activated clonotypes? The simple question is, okay, you identify 100,000 clonotypes. What are you going to do with 100 clonotypes? Nothing, really. You're interested probably in the most interesting clonotypes. For example, clonotypes which are activated. And here's the example of the vaccination studies, what we've done. And I was Guinea pig, which actually provide all these blood samples for this experiment. And you can see here two types of vaccination. One of them is trickle vaccine, tetanus, diphtheria, and pneumonia on the top, and Shingrix vaccine. And you see we collected multiple time points before vaccination and after vaccination. And we ran expression profiling and immune repertoire profiling studies. If we just ran genome-wide expression profiling for the samples, we identify set of the signaling pathways which are approximately at 2 days after vaccination, activated bunch of the innate immunity pathways. At approximately 1 week after vaccination, we have activation of adaptive immunity pathways. Okay, so we identify some markers which are activated at different time points. What's happened with the immune receptors? And here's the both vaccinations, rather complex slide. And let's look first on the right side. This one is IgH, it's heavy chain of immunoglobulins or B cell receptors. And you can see here for both vaccination, at approximately 1 week after vaccination, we have strong proliferation of the B cells. And you can see it's if you use DNA as a starting material for this type of expression profiling, for RNA we can see very matched set of the clonotypes identified at the RNA level. I would like to put your notice that RNA is significantly more sensitive, approximately 1,000-fold than DNA, in detection of activated clonotypes after vaccination for both vaccines. If you look at the T cell receptors, you don't see really significant proliferation when we use DNA sample, for example, as a starting material, but we can see still activation of T cell receptors in the T cells. You can see it's approximately the same time, about 1 week. It's not so significant as for B cells, which is rather obvious because here probably we can see mainly expression from the plasma cells, which actively produce antibodies after vaccination. So, looking at this data, you can speculate that we can identify activated clonotypes by simply comparison between RNA and DNA data, by normalization of RNA data to DNA data. In this case, you identify activated clonotypes. In this type of the experiment, which is longitudinal studies, probably it's not very critical because you just have time course and you can see which one of the activated. But if you have single sample, for example, blood from the cancer patient, very often you don't have time course. You have only single sample, and this strategy can be used in order to identify activated clonotypes. Couple words about connections between sequence of T-cell receptors and antigens. We try to really match T-cell receptors which we identify in these studies with the potential antigen which induce this type of the immune responses. We collaborate with the company ImmuneWatch in Europe, and these guys one of the best who have developed special machine learning algorithm detect which allow it to match these sequences. And for first vaccination, they were not able to discover any hits, but for second vaccination, they show that majority of clonotypes which we identify in these studies correspond glycoprotein E of zoster virus, which is integral part of this vaccination. So it's if you generate any of this type immune receptor profiling data, I encourage you just maybe to contact these guys, and they will try to really to find what is a potential candidate induced connected to this T-cell receptor. How to characterize? Next question. Okay, you identify intrinsically activated clonotypes. What is the type of the cells correspond to these activated clonotypes? What is the phenotype? And definitely traditional technology for this type of the studies is to do 10x genomics or parse or scale biosciences or REPSCI platform, where you just encapsulate cells in microvells or in droplets and generate a single cell expression profile from each cell, like shown here on the top of this slide. After that, you group the cells together based on similarity and expression profile in some sort of the clusters using t-SNP plot or any other strategies. And after that, you connect each cluster to specific cell type using expression of specific markers which are critical, for example, specific for naive cells, memory cells, effector cells. And you generate both immune receptor profiling and expression profiling for each of these group of the cells. It's actually the similar strategy can be done very similar if you just sort the cells based on the expression of the same cell-specific markers, and you can generate expression profiling and immune repertoire profiling using technology that I discussed previously. And another strategy, what you can do, you can sort cells in 96-well plate and do similar assay where in each well you can put primers for immune receptors and for some marker genes which are critical to characterize phenotype of the cells. So here's an illustration how it can be done, for example, for cell fractures. You sort cells for different naive memory effector cells, CD4, CD8, and do both expression profiling and adaptive immune repertoire profiling. And you generate data like shown here on the next slide. For each cell fractions, you get set of the immune receptors and key markers which correspond to these specific cell fractions which allow you to characterize phenotype of these cells. Similar strategy if you just analyze small number of the cells, and here's some application which is very typical. For example, if you try to identify activated cells induced, for example, by specific peptide or specific antigen. Usually when you try activate PBMC with any antigen, number of the cells which you activated is very small. It's at the level it's maybe a few hundred. It's very rare you can see thousands of the cells. In this case, what you can do, you can just sort cells when you analyze the activated PBMC cell. It's simple. You can sort the cells directly in 96-well plate. In each 96-well plate, we have pre quoted set of primers for immune receptors and for marker genes. And you can run just simple assay, like I described right now, and generate similar set of the data where you have chain pairing, identification of, for example, alpha and beta chain, and set of 30 different markers, top markers for T cell or B cell. And it can be applicable, for example, for if you sort cells based on expression of activation markers like CD137, CD69, CD25. Or if you try to label the cells using MHC tetramers or dextramers, or if you try to sort some specific fractions based on the expression, for example, of exhausted cells. And couple words about, in general, where you can use different technologies. We need to realize that bulk profiling using some RNA or DNA from the samples is the most cost-effective technology. It's no doubt about it. For example, and second, it doesn't require alive cells, which is very critical. So it can be some cells, PBMC or tissue samples already completely fixed or even put in some sort of the Tempus test tube for storage or PAX gene test tubes. So cells are already lysed and it's not necessary to be alive. So cost of bulk profiling is very low. It's something like $50 per sample, and you can run 96 or even 200 samples in one day and get quantitative profiling data. And so that's why it's cost effective and easy to do. As soon as we talk about single cell profiling, you don't have any quantitation. We need to realize that very clear. No quantitation data in single cell data if you use less than 1 million cells per sample. And 1 million cells per sample, it's already extremely expensive. If you use 10x platform or parse, forget about it. You're talking about $10,000-$20,000 per sample. Yeah, for bulk profiling, we are talking about $50 per sample. It's make big difference. And so that's why, but combination, definitely bulk profiling don't give you chain pairing information. If you need to characterize and identify interesting clonotypes, bulk profiling is the way to go. If you want to really to do more deep analysis, to get full-length information, chain pairing information, single cell analysis probably it makes sense already to consider, and it will be the only way to do it this status. And I would like to stop right now at this moment and just summarize what we discussed today. We discussed today development of air immune receptor profiling assay, which a really cost-effective strategy for the bulk profiling in the hundreds of the samples, and give you comprehensive quantitative information of presence of different clonotypes in biological samples, including blood, any tissue samples, which doesn't require purify specific even T or B cell fraction. You can directly work with the tissue sample, RNA purified from tissue samples, and still get the same information. It's also we discuss strategy for detection of antigen-activated clonotypes based on normalization of AR RNA to AR DNA data. We discuss strategies for functional characterization of the cells based in the cell fractions or at single cell level using cells sorted in 96-cell plate. And all these technologies available as the kits or custom services. If you're interested really to use this technology, please welcome us and visit our booth 509